preparing to stream.
Okay, we are live. Okay, so hi everybody. Thanks for joining our webinars, our DevMech webinars. Today we have the great pleasure to have with us Srikala Raghavan. And Srikala joined the University of Cambridge for a PhD and did her postdoctoral training with Prof. Elaine Fuchs at the University of Chicago and later on at the Rockefeller University. In 2005, Srikala established her lab at Columbia University in New York at the College of Dental Medicine and Department of Dermatology. Then in 2012, she was recruited to INSTEM in Bangalore. INSTEM stands for Institute for Stem Cell Science and Regenerative Medicine. And there she established the Center for Inflammation and Tissue Homeostasis, where she's now an associate professor. At the same time, in January 2020, she started a joint appointment as principal investigator at the Skin Research Institute in Singapore at A-STAR. So, I mean, she had a great career, and I think she's extremely proud also, and we are really I was excited to read her paper on science the last November. It's a paper she published together with Valery Vasyukin, and talking about the regulation of junctional strength, especially looking at tricellular junction. So I'm looking forward to your talk about junctional instability that can override intrinsic quiescent of stem cells. Thank you, Srikala.
Thank you, Christina. Thank you, Andrea. Thank you all for being here today. And I guess good morning to all of you. It's the night here in Singapore. But as Christina mentioned, I'm going to talk to you about the junctional instability and its role in maintaining stem cell quiescence. Okay, let me see. Okay, so in my lab, we use the murine skin as a model system to understand and address questions dealing with epithelial homeostasis and immune regulation at barrier surfaces. And one of the reasons the skin is a really exciting system is because, of course, it is the most external organ that we have. Let me just turn on our laser pointer. Sorry. Laser pointer. There you go. Okay, so it is our most external organ. It also is our largest organ. And it also constantly turns over in development. And so the skin comprises of an external part called the epidermis and then a lower part called the dermis. In development, the epidermis is ectothermally derived and the dermis is derived from the mesoderm. The epidermis and dermis are separated through this acellular part called a basement membrane, which is comprised of extracellular matrix molecules. And in addition to the epidermis and the dermis, the skin also comprises of multiple appendages, the main one being the hair follicle. And the hair follicles is closely associated with the sebaceous glands. And of course, there's also the sweat glands. And so there's been a huge amount of interest in the skin, not only because, of course, it's sort of, your outermost organ, it's the largest organ, it constantly regenerates, but it's also chock full of stem cells. And the interest in long-term stem cells sort of really arose from really phenomenal work over the past 25 years, looking at these quiescent stem cells within the skin compartment, which are associated with a compartment called the bulge. And so, as I said, the skin constantly turns over, the epidermis turns over every 3 weeks. And these are sort of replenished by pools of stem cells. So there is the epidermal stem cells, which reside primarily in the basal layer of the epidermis. And this is the only dividing cells within the skin layer. Everything else is postmitotic. And then within the hair follicle itself, there are several pools of stem cells. And there's been sort of a lot of interest, as I said, over the past several years in trying to understand the properties of these stem cells. And work from multiple labs have identified several intrinsic factors, which are all listed here, that are important for either the specification or the maintenance of these stem cells. So an interesting thing about a hair follicle is, and that is true both for the humans, and also the murine system that we study, is that all the hair follicles that you are ever born with are specified primarily in embryogenesis. And once they are specified, the hairs undergo a process called hair cycle, wherein a hair shaft a new hair shaft grows out, and then it replenishes the old hair shaft. So the old hair shaft falls out. And so this process is cyclical, and it goes through a growth phase known as anagen, a destructive phase known as catagen, where the 2/3 of the growing part of the hair follicle gets destroyed, and then a resting phase called the telogen. And there is also a signaling center that's associated with each hair follicle, and this is called the dermal papilla. And this is the interactions between the hair follicle, the stem cells, and the dermal papilla are thought to be really important in the cycling process. And so while all hair follicles are specified in embryogenesis, the cycling itself takes place throughout the adult life. The first 2 hair cycles in mice are synchronized, meaning every single hair follicle does the same thing. And subsequently, the hair follicles become asynchronous. And this will become a little bit important later on in my talk. And so, as I said, there's been sort of a huge amount of interest in trying to understand what the various factors are that specify or are important for the maintenance of these stem cells. And most of these are transcription factors. Some of them are cytoskeletal proteins. And there's also this really interesting cell surface marker called CD34, which is a hematopoietic marker, which is also expressed within the bulk stem cell compartment. And so while we know quite a bit about cell intrinsic factors, we know much less about what the role of the niche might be in maintaining the stem cell pool. And so whether it is cell to substratum junctions or even cell to cell junctions in maintaining these. And so we sort of got very interested in this idea of what these factors, how the cell to cell junction might be playing a role in maintaining these. And so I'm just going to introduce you to the cell to cell junction that we have been looking at, which is primarily the adherence junctions. So the cell junctions in the stem cell compartments, which are primarily keratinocytes, come in 2 flavors. They are the desmosomes, which are these keratin-rich, plaques that you find. And then in between the desmosomes, you have the adherence junctions. And the adherence junctions are, of course, nucleated by homophilic cell adhesion molecule ecadherin. And then it has a variety of accessory molecules or cytoplasmic associated proteins, the catenins, which then through this protein called vinculin associates to the actin cytoskeleton network. And so while it's very difficult to look at an EM or to capture these, what's known is that these junctions form between 2 desmosomal structures. And so we were very interested in trying to understand what the role of these cell junctions would be in the epithelial stem cell niche, specifically in this process from where the hair follicles start the process of growing from an otherwise quiescent phase and throughout the cycle of the hair follicle. And so to start off with, we decided to look at what the epithelia actually look like within the growing follicle and within the stem cell niche. So just to orient you, these are growing hair follicles. So this is sort of the bulge compartment, which is your stem cell compartment. And this is now the newly growing hair follicle. And one of the properties of the bulk cells is that they are quiescent, so they don't really divide much. And we know that because now if we actually label these cells and follow the label, they are label retaining, and then they don't actually lose their label, suggesting that they're very slowly dividing cells. And so in a growing follicle, what we were able to see was that the bulge compartment itself was not labeled with proliferation marker Ki67, but as the follicles were growing out, they started picking up the Ki67 or the proliferation marker. And so when we went back and looked at the junctional proteins, primarily ecadherin, alpha-catenin, and vinculin, what we could see was a very strong association with of these proteins in the bulge compartment. So there's a very strong expression. And then as these follicles were starting to grow out, these junctions or this expression was reducing. And so when we look at this in EMs, what you can see is that as the follicles are growing out, you see fewer and fewer cell-to-cell junctions in between the growing part of the hair follicle. And this is in stark contrast to the actual stem cell compartment in a quiescent hair follicles compartment. And so in this compartment, what you can see is that there are no proliferating cells, as you can see. And also there's a very strong association of these junctional proteins with the actin cytoskeleton network. And I would mention that the actin network also looks pretty organized and cortical in nature. And if you go back and look at what the junctions look like, you can see, and this is just marked by the red asterisks here, what you can see is there are way more junctions. So what this told us was that there seems to be a change in the number of junctions and sort of the quality of junctions as these cells are proliferating and as the hair follicles are growing out. And so I wanted to ask, what might the role of these junctional proteins be? Because clearly they seem to be getting regulated in terms of their expression. So what might the role of these junctional proteins be within the stem cell niche itself? And to do this, we decided to focus on this protein called vinculin. So vinculin is an adaptive protein, and it comprises of a head and a tail domain, which is joined together by a hinge region. Vinculin is a mechanotransducer, and I'll talk about that in the next slide. But in fact, it exists in two conformations: in a closed conformation where the head and the tail are bound to each other, and then in an open conformation. And the unfolding of vinculin itself requires force. So within the epidermal keratinocytes, which is the cell type that we're interested in, vinculin is associated with two specific junctions. So it is associated with the cell-to-cell junctions as seen here, which is the adherence junction. But vinculin is also associated with the focal adhesions, which are the cell-to-substratum junctions, which are just marked over here. And so, as I mentioned, vinculin is a mechanotransducer. And what we mean by that is that vinculin, in a force-dependent manner, is able to bind to this protein at the level of the cell substratum junction called talin. And this allows for talin to unwind or unfold. And then this unfolding of talin gradually recruits other proteins and facilitates its association with the actin cytoskeleton network. And so all this is happening in a force-dependent manner. And likewise, at the level of the adherence junctions, vinculin associates with this protein called alpha-ketinin and allows for, or facilitates its unfolding. And then this in turn is able to bind to the actin cytoskeleton network. And the other thing that vinculin brings in is also multiple actin remodeling enzymes. And so this also helps to sort of reinforce the forces that are generated by these junctions. And so what might the function of vinculin be in the stem cell compartment? So to address this, we generated a conditional knockout of vinculin in the skin, and we used a skin-specific promoter, which is the keratin 14 Cre-driven knockout. And so to our surprise, the animals were actually completely fine. So if you remove the function of vinculin in the skin, you get animals in the correct Mendelian ratio. The only thing that we could see that was a little bit different between the control and the knockout was that these seem to have less or sparser hair. Now, even for those who are uninitiated, I think this is very clear. You can see that the hair coat looks a little bit sparse. And so this is just the analysis to show that we truly have knocked out vinculin in vivo in the hair follicles, in the keratinocytes, and of course in Westerns. And what was also interesting was that this sort of patchy or less amount of hair seemed to sort of follow. So these animals were about 50 days old. And if you take animals that are a year old, they still don't seem to get a full complement of a hair coat. And so that was, as far as we could tell, the only overt phenotypes that we could see in these animals. And so because this was this hair kind of phenotype, we were interested in understanding what exactly is happening. And so what we decided to do is to take animals for the first 60 days. And this covers, as I remember, I told you the hair follicle goes through these cycles of growth. So this is the growing phase, this is the regression phase, and then this goes back to the telogen, which is the resting phase. What's really fascinating about this is the incredible change in size. So you know the follicles are undergoing about a 10-fold increase in size, and then they're retracting back to their almost their original size. This is sort of your scale bar over here at 100 microns. And so what we decided to do was to go back and look very carefully at what is happening with the hair follicle. And immediately what we could see was that the hair follicle cycle in the vinculin knockout looked pretty perturbed. For one thing, the hair follicles didn't seem to grow out as much. For another thing, it just didn't seem to follow the correct cycle. So it didn't seem to grow for the same number of days. It seems to retract much earlier and then got into this sort of telogen stage much earlier. So if you just simply look at the outlines of the follicles, it becomes much clearer. And what we were able to show was that the hair follicles in the knockout were never reaching the size of the wild type. They seem to be in completely different phases. So this just marks anagen, catagen, which is the destruction phase, and telogen, which is the resting phase, or the, so you could see that they were in completely different phases. And there seemed to be a decoupling between the size of the hair follicle and the size of the skin. So what is interesting is that the size and thickness of the skin increases with the size of the hair follicles. And in this case, you had, there was sort of a decoupling in this. And so what this told us was that we followed this for 2 hair cycles, and this is just summarized here. And what we could show was that in the first hair cycle, there was a longer catagen, so a longer destruction phase. And then immediately this sort of went into the growth phase, a catagen, and sort of the next telogen at postnatal day 35 or 37, whereas in the wild type animals, this only occurred after day 100. So this is now telling you that this cycle was extremely accelerated in the conditional knockout of the vinculin. And so what I've told you so far is that the knockout animals are fine, but they have sparse hair and accelerated hair cycle. They don't seem to really acquire this quiescence phase. And of course, the hair follicles are much smaller. So, how might the loss of vinculin, which is, this adaptive protein that seems to be associated with focal adhesions and adherence junctions, how might the loss of this explain these effects? And so, of course, one could argue that the phenotype could be both either driven through the focal adhesion function or the cell substratum function of vinculin, or the adherence junction function of vinculin. And so we looked at this long and hard. And in fact, what we were able to determine was that, in fact, the cell substratum role of vinculin didn't play much of a role in maintaining these stem cells. And in fact, what was important was the adherence junction function of vinculin. And so this was sort of, this is what we focused on next. And so this is just sort of a 3D rendering of the adherence junction again. And so this is your plasma membrane, your cadherins, which are transmembrane, binding to scatenin, so both P120 and beta-catenin. And then beta-catenin brings in this protein called alpha-catenin. And alpha-catenin, as I told you before, lives in this sort of folded state, and it requires the recruitment of vinculin to unfold. And so when vinculin binds to alpha-catenin unfolds, and then is associated with the actin network or exposes actin binding domain and binds to the actin cytoskeletal network. Likewise, vinculin also has an actin binding domain. And so the binding of vinculin is critical for the unfolding of alpha-catenin. And so we then asked, okay, so what happens when you sort of remove, what happens to these junctions and the adherence junctions when you remove vinculin? And so we started looking at the adherence junctions within these hair follicles itself. So we decided to focus both on the first telogen phase, which is the first time that the hair follicles become their shortest, and then the second telogen phase. And what I should note is that while the first telogen phase were more or less at the same age, the second telogen phase, of course, was at a completely different stage because these hair follicle cycles are accelerated. And so we were really looking at the size of the hair follicles to determine that these were really in telogen. what was immediately apparent when we looked at the junctions was that the junctions were quite perturbed. So this is just looking at the expression of E-cadherin. And what you can see was that the expression of E-cadherin was quite messed up, as was the expression of alpha-catenin at these junctions. And this was further corroborated when we looked at EM images of these junctions. And what we could see was normally in the wild type, you would get these beautiful sort of cell-to-cell junctions, and you would see these very nice desmosomal structures, and you can see in these higher magnification insets. And in the knockout, what we found was that there were these gaps or sort of these cells that were not being held together. And it almost seemed like the two membranes were being pulled apart in the knockout. And what we also saw was that the number of desmosomes, which are these cell-to-cell junctions, which in some sense we're using as a proxy for the adherence junctions, were significantly reduced in the knockout hair follicles, but not in the interfollicular epidermis. So this is, of course, vinculins also expressed in the basal cells and in the interfollicular epidermis, and that didn't seem to have any issues. It's only in the hair follicles that we found problems. And so we also wanted to look at the adherence junction formation in keratinocytes that are derived from these pulse stem cells. And so what we could show was that when you take these keratinocytes and allow them to form junctions in culture, the wild type form these beautiful adherence junctions, which are nucleated both by E-cadherin and alpha-catenin. And in the knockout, what we could see was that these junctions were very, very perturbed. As you can see here, these cells fail to zipper up. And so when we do a scanning EM, becomes very clear. So this is just the final stages of zippering up between two adjacent keratinocytes. And what you can see is these beautiful phyllobotial structures that are going from one cell to the other, which aid in the zippering. And in the knockout, what we could see was that, in fact, the zippering is quite perturbed, and these phyllobotial structures, in fact, don't appear normal at all. And so clearly there was a problem with forming normal adherence junctions. And so to look at this more carefully, what we decided to do was to just look at live cell imaging. So these are wild type keratinocytes that have been transfected with an E-cadherin GFP and just allowed to form junctions. And so what you can see in the wild type cells is that, pretty soon after the two cells meet, they form this really nice stable junction, and they retain this junction for the duration of this observation. And in fact, when we follow this through, they can keep these junctions together for long periods of time, in excess of 18 hours. The knockout cells, on the other hand, firstly, they were much smaller, as you can see. So this is the same magnification. But the other thing was that these cells just failed to maintain stable junctions. So if you follow this labeled cell, what you can see is that it keeps on making and breaking junctions quite quickly. Again, suggesting that there was sort of an issue with maintaining stable junctions. So we have already talked about, we've looked at the role of vinculin and said that, in fact, when you remove this, the junctions start becoming unstable. But that led us to ask the question, within the adherence junction, there's also this other protein called alpha-catenin, right? And so what is the role of alpha-catenin in this process? And what might alpha-catenin be doing at the adherence junctions vis-a-vis junctional stability of the adherence junctions in the stem cell compartment? And so to address that, we collaborated with my good friend Valerija Vasiukin, who generated the conditional knockout of alpha-catenin. And I think what you can see right away is that the alpha-catenin knockout, in fact, does not have any hair follicles. So clearly the phenotypes here are more severe than what we see in the vinculin knockout. And this is just to show that we've knocked out alpha-catenin just in the stem cell compartment. In fact, alpha-catenin, if you knock it out within the skin globally, then these animals are lethal because they don't form any adherence junctions. And so just knocking it out in the stem cell compartment was enough for these animals to lose their hair. And then if you go back and look at what is happening with the hair follicles in these animals, what we could find was very similar to vinculin. So the hair cycle is a little bit delayed in the alpha-catenin knockout because this promoter knocks out alpha-catenin at postnatal day 6, as opposed to the embryonic day 14, which is what happens with the K14-driven knockout. Nonetheless, once these animal hair follicles go back into telogen, they fail to grow back a normal anagen. So again, the hair follicle is cycling faster, and they basically fail to form these long hair follicles. The junctions themselves are quite perturbed. So if you go back and look at the expression of E-cadherin in these hair follicles, you can see, as opposed to the wild type, the expression of E-cadherin is highly perturbed. And of course, these are just the formation of junctions. You can see, of course, the alpha-catenin knockout has no alpha-catenin, and vinculin is no longer expressed at cell-to-cell junctions, because of course, vinculin and alpha-catenin are bind to each other in order to be recruited to the cell-to-cell junctions. And so, so the next question that we asked was, of course, these junctions are not forming properly. So why are these junctions weak, and what are the forces that are being generated by these junctions in the knockout cells? And so to get at this directly, we decided to collaborate with Yan Ji at the Mechanobio Institute and his postdoc Shihai. And what they had generated was this pretty cool sort of bead capture system, wherein what we did was we took 3-micron beads and coated it with E-cadherin, and then attached it to the surface of cells, both wild type and knockout, and then captured these beads with a micropipette. And once the beads were captured, we basically moved the stage, so moved the cell away from the micropipette. And so we were then able to calculate sort of the detachment force that is required for the bead to be detached from the cell, which is both a function, of course, of the strength of the junction itself, but also, of course, the bending constant of the pipette. And so what this gave us was really a direct measurement of the forces that are experienced by this cadherin coated bead when it is attached to the cell surface of either a wild type or a knockout cell. So this is just the force measurement. And so the detachment, so the amount of force that it takes to detach this 3-micron bead from a wild type cell is approximately 6 nanonewtons. And from the knockout cell, you can detach the bead with about 2.5 nanonewton force, suggesting that these junctions are severely compromised. And if you add back vinculin, so in this case, we added back vinculin as a vinculin GFP, we're able to completely rescue this adhesion force, suggesting that the loss of vinculin was what is responsible for the loss in the adhesion force here. And this is just to show you what happens in these cells. So this is the control cells, and you can see that vinculin is expressed both at the junctions and at the focal adhesions. Alpha-catenin, of course, is expressed only at the adherence junctions. In the vinculin knockout, very interesting, there's a huge amount of alpha-catenin that's expressed. However, even though alpha-catenin is strongly expressed, it's not able to generate forces. And when you now put back vinculin GFP, you're able to restore the junctions as well as the forces. So what was also really interesting was that it's not as if these junctions were not expressing junctional proteins. If anything, the vinculin knockout cells were expressing higher levels of both cadherins as well as alpha-catenin. In fact, almost 2-fold higher levels. So it's almost as if the cells is compensating by putting out extra protein, but it didn't matter. Even though there was more protein being expressed, the junctional forces were much weaker than the wild type. And so we also then measured the forces in alpha-catenin knockout cells using the same methodology. And so I'll just walk you through this. So this is just an alpha-catenin control where you can see that alpha-catenin is at the adherence junction along with vinculin. And then in the alpha-catenin knockout, of course, vinculin does not make it to the adherence junction. So it's only present at the level of the focal adhesion. And then when you put back alpha-catenin, you're able to rescue back the junctions. And so I'm just going to take you through the force measurements. And so in the control, you see that it's about 5.5 to 6 nanonewtons of force to detach the bead. When you put back full-length alpha-catenin, you're able to rescue it. And when you, in the alpha-catenin knockout, again, there's the level of force is only about 2.5 nanonewtons. So we then asked the question, what is the role of vinculin in this process? And so what we decided to do was to rescue the alpha-catenin knockout cell with a version of alpha-catenin, this is the L344P mutant, that no longer is able to bind to vinculin. Okay, so this is in some sense, even though alpha-catenin is expressed, it can no longer bind to vinculin. And so this is just shown here. So this is the L344P rescue. And in this case, this is in green. So you can see that alpha-catenin comes back to the junctions, but vinculin is no longer recruited to the junctions. It's only found in the focal adhesions. So under these conditions, what we were able to show was that, in fact, when we rescued the alpha-catenin with a non-vinculin binding version, this was also not able to rescue the junctional forces. Again, suggesting that in fact it's vinculin, the presence of vinculin and its association with alpha-catenin that's absolutely critical for maintaining forces that are generated by these junctions. And so what I've told you so far is that in the wild type bulk stem cells, they have normal cell junctions in vivo and in vitro and have a relative, so in vitro, they have a relative force of 6 nanonewtons. And what I mean by that is the detachment force in these bead experiments that we did. And the knockout bulk stem cells have abnormal junctions in vivo, and in vitro, they have a relative detachment force of about 2.5 nanonewtons. So what are the consequences of having these weak junctions now? Because of course the proteins are all there, but the junctions are very, very weak. And so to understand this, what we decided to do is to just do an NGS analysis of the isolated bulk stem cells. So this is just showing you that we've been able to isolate using the previously mentioned CD34 marker. And so when we isolated the cells and subjected them to an NGS analysis, what we found was one of the most upregulated pathway was the targets of the hippo-YAP signaling pathway. And so the hippo-YAP pathway is a pathway that responds exquisitely to mechanical signals. So YAP is a transcription factor that translocates to the nucleus and is very important for turning on its targets, which include cell cycle genes such as cyclins. And it can be controlled both at the level of cell-to-cell junction, cell-substratum junctions, so at the level of focal adhesions. So the localization of YAP to the nucleus is explicitly controlled by cell substratum junctions, as well as cell-to-cell junctions, as well as cell geometry. So when a cell is less well spread, YAP translocates to the nucleus, and when a cell is highly spread, then YAP translocates out of the nucleus. And so this is, the nuclear translocation of YAP is a very good indicator of the mechanical status of the cell, which expresses it. And so given that the targets of hippo-YAP signaling were upregulated and YAP itself was upregulated in our knockout, we wanted to find out what the status of YAP is. And so this is, again, we looked at the quiescent stem cells. So in the normal quiescent stem cells, what you would see is that YAP is primarily associated at the membrane, and it nicely co-localizes with the phalloidin, which is as the plasma membrane. In the knockout, what we could see was strong nuclear localization of YAP. And this was true in keratinocytes and culture as well. So, and under conditions of very high confluence, which is a similar status to what you would see in these bulge compartment, you see that YAP is excluded from the nucleus. But in the vinculin knockout, under conditions of very high confluence, YAP was primarily located in the nucleus. And this is just the quantification of the YAP nuclear localization. And so one of the consequences of having high levels of nuclear YAP was that we actually lost the long-term label retaining cells. And so, as I mentioned before, these cells express the cell surface marker called CD34. And so in the first telogen, we already started losing CD34 expression. But by the time these animals have the second telogen, there's a complete loss of the stem cell marker. We also lose the long-term. Remember, I told you that these cells are quiescent and have a long-term label retaining. So you can actually label them with BRDU or EDU and chase them, and they will retain the BRDU expression, both in the first and the second telogen. And the vinculin knockout cells did not have any BRDU labeling. And in fact, these cells, because they're quiescent, do not proliferate. And so we do not see any Ki67 positive cells in the first and second telogen of control follicles, but the knockout follicles are full, chock full of proliferating cells. So the loss of junctions and the increased nuclear localization of YAP triggered sort of this loss of label retaining and long-term stem cells in the vinculin knockout. So what is the mechanism that underlies this, right? So because of course, in the vinculin knockout, you have high levels of alpha-ketene and you have high levels of ECadherin. So what is it that then triggers this nuclear localization? So it turns out that in fact, alpha-ketene is one of the proteins that can sequester YAP in the cytoplasm in a density-dependent manner. So alpha-ketene binds to YAP through its association with this adaptive protein called 1433. And of course, when YAP is in its phosphorylated state, it is in the cytoplasm, and then when it gets dephosphorylated through a protein phosphatase called PP2A, it can translocate into the nucleus. And so because of this finding, and this was also my collaborator Valera was involved in this study, we were very interested in understanding the relationship between alpha-ketene and YAP in the knockout. Now remember, there's a huge amount of alpha-ketene that is being expressed. So the question that we asked was, why isn't alpha-ketene then able to sequester YAP1 in the vinculin knockout? And so when we went back and looked at what is happening with alpha-ketene, so remember I told you that alpha-ketene needs to be in this open conformation in order for it to be contacted by vinculin. And so there is a conformation-specific antibody called the alpha-18 antibody that recognizes this open conformation. And so in the normal, in the control bulge, you can see that there's a beautiful expression of this alpha-18 isotope. And there's a very nice concordance with the alpha-ketene and with this alpha-18. When we looked at the knockout, on the other hand, what we could see was huge amounts of alpha-ketene, but virtually no expression of this open conformation antibody. And this is sort of quantified over here. And so we went back and looked at this more carefully in cells in culture as well. And so as these junctions are forming, you can see this beautiful expression of alpha-18, which is the open conformation antibody at the junctions. And the knockout cells, even though they express huge amounts of alpha-ketene, fail to express this conformation-specific alpha-18 or the open conformation of alpha-18. And so when we looked at this more carefully using 3D SIM, what we could see was that, so the alpha-ketene is in green and the actin cytoactin is in red. And what we could see is a beautiful capture or a co-expression of alpha-ketene with the actin cytoskeleton network, whereas in the knockout, we could not see this happening at all. So suggesting again that in the open conformation was simply not present in the vinculin knockout. And so what we were also able to show was that in fact, it is this open alpha-ketene that is able to sequester YAP. So if we were to carefully look at where YAP is expressed with respect to this open conformation antibody, we could see that there was a very nice and strong localization of YAP with alpha-18. Whereas in the knockout, we could see that the alpha-18 conformation antibody was not expressed, and there was a nuclear localization of YAP. And when we look at cells in culture, when we sort of rescued, we took knockout cells and rescued them with vinculin GFP. So this is a colony of cells that has been rescued. And these now re-express alpha-18 on their cell surface. And in fact, these are the cells that now start precluding YAP from the nucleus. Again, suggesting that, at least in our system, one mechanism by which YAP seems to be sequestered to the plasma membrane is through the presence of this open conformation of alpha-ketene or the presence of alpha-ketene itself. And so the other thing we looked at is because we know that alpha-ketene is associated at the plasma membrane through this linker protein called 1433. So we very carefully analyzed the expression of 1433 and YAP. And what we were able to show was that in control follicles, there was nice concordance in the expression of 1433. But in the knockout, the expression of 1433 was very diffuse, not necessarily associated with the plasma membrane. And of course, there was increased nuclear localization of YAP. And so what we're left with is this model where we're in the control cells, we have cell junctions that are nucleated to cadherins and catenins. And in the presence of vinculin, there is this stretched form of alpha-catenin. And of course, both vinculin and alpha-catenin are very important to contact the actin cytoskeleton network. And the stretched form of alpha-catenin, through its interaction with the adaptive protein 1433, keeps YAP at the plasma, at the level of the plasma membrane. In the absence of vinculin, what happens is that the alpha-catenin folds on itself, and 1433 is then released from this from alpha-catenin, and then YAP also is released, and YAP then goes into the nucleus. We have actually been able to show, I've not shown you data here, but we do see decreased levels of phosphorylated YAP. It could be expected because now YAP is translocating into the nucleus. And once YAP translocates to the nucleus, it basically turns on its target genes, which include cell cycle regulators, which is why we see increased amount of proliferation in these vinculin knockout cells. And so to summarize what I'm telling you, what I've told you today is that the wild type bulk stem cells have normal cell junctions and they are contact inhibited. So if you look at these, this is very reminiscent to us of contact inhibited cells, particularly the way the actin cytoskeleton network is arranged. These cells, of course, are quiescent and they have cytoplasmic gap. And in the knockout bulk stem cells, you have abnormal cell junctions. These are not contact inhibited, non-quiescent, and there's an increased nuclear localization of YAP, which provides a mechanism for increased proliferation. And so what our study can conclude then is that the reduction in the intracellular junctional forces is sufficient to sort of override the intrinsic quiescence that is present in these stem cells. So of course, we looked at the markers that are expressed in these stem cells, and all of these are normally are expressed. So these stem cells do express the markers, but just the failure to maintain these normal cell-to-cell contacts is enough to pull them out of quiescence. And so these stem cells basically constantly divide, and that's what explains the constant cycling of these hair follicles in the vinculin knockout. And so I'll end there, and just remains to thank the people involved in this work. So all of the work I talked to you about today was done by a fantastic graduate student, Ritushri Biswas, who's here, and an incredible postdoc, Abhinanda Banerjee, who's here. So these were the dynamic duo that really drove this project. It was also started in my lab by Sergio, who was an exchange student from EFOM in Milan. This is the rest of my lab. And then my lab in Singapore says very lucky to be able to recruit Abhinanda here. And so she was here with me. And then there's Ryan and Shimin. My collaborators, so Raghavan, who was just helped us with all of the NGS analysis, and Shihai, who was from Yanji's lab, who helped us to do all the mechanical experiments, mechanobio experiments. Other collaborators, including Yanji, Marius, who provided us with all the YAP antibodies and fantastic discussions. All my funding sources, and thank you for your attention, and I'm very happy to take questions.
Thanks a lot, fantastic talk. Really enjoyed it. I think there are many questions, but maybe while people warm up and think about, I'd just like to ask you a very general question about the system. So you seem to assume that there is the major role for the growth and the shrinkage of the follicle is due to cell number, so proliferation and maybe apoptosis. I don't know if you mentioned about that. But is there also some sort of cell migration / invasion by the cells at the tip that are pulling the follicle?
Right, that's a really interesting question. So, so what happens here is that it is a combination of, definitely a combination of cell proliferation So what we know is besides, we know a lot about the signals that drive this, right? So the initial signal that drives this, this sort of quiescent to growth phase that I was talking to you about is a Wnt signal. And that's very interesting because, Wnt signals or Wnt or, the effector protein is beta-catenin, and it was just conveniently present right there at the junctions, right? And so, so the initial signal is a Wnt signal, and then the subsequent signal is hedgehog. And so there is, a very strong proliferative push that drives this. You were asking about cell stretching. Is that right, Andrea? Is that what you?
No, actually, I kind of have two questions. So during the shrinkage, is that then driven by apoptosis?
Yes.
And if during the growth, there is a role also for active cell migration, so like invasion of the cells. I think the papilla cells are called. I don't know. I forgot it.
The dermal papilla. Yes.
Dermal papilla.
Yes. Yes. So yes, so I think that's also, yes. So to answer your first question, yes, the catagen phase here is driven by apoptosis, and one of the ways that cells are cleared is also through entosis. So there's a lot of engulfing of one cell by the other. My good friend and earlier colleague Valentina Greco has done some very beautiful studies looking at live imaging of these processes. And if you get a chance, you should look at some of the movies she's generated. In terms of the invasion, I think you're absolutely right. In fact, what I love to refer to the growing hair follicle as a controlled invasion. So in fact, what the hair follicle is doing is it's actually growing into area of very dense and rich extracellular matrix. And it has to actually carve a path through that. And then later on, as the follicles grow, it also is getting through sort of this adipocyte layer. And yes, there are lots of MMPs and other things that are expressed as hair follicles are growing down. And in fact, that's one of the other things that the lab is focused on, because we're very interested in understanding the role of matrix remodeling enzymes in this process.
Right, that's very nice. Very nice system.
May I?
Christina? Yeah. You may.
Thank you. Srikala, fantastic. Thank you.
Thank you.
Okay, I have two questions. One is about the fact that vinculin can also bind to beta-catenin, right? So I'm wondering whether, have you ever tried in an alpha-catenin knockout system to rescue with the open conformation of vinculin to see if it's possibly binding directly to a beta-catenin layer? And if that would rescue.
So our reviewers asked us this, so we were never able to find, so in the alpha-cat knockout, I guess both E-cadherin and beta-cat are also displaced, right? So there's nothing to really hold vinculin there. You're absolutely right. It could come directly through beta-catenin. But we were not really able to find, so they did actually ask us to put vinculin there in a non-alpha-catenin dependent manner.
Yeah.
And we try as we might, we weren't really able to find a good construct for that.
I see.
To look at that. And I don't know if you guys have something, then I would love to test that out.
Yeah, we'll talk about that later.
Yeah, we can talk about that. And the question is whether one can completely bypass alpha-cat in this process. But remember, in the vinculin knockout, there's a ton of beta-catenin, alpha-catenin, and E-cadherin, right? And so it doesn't matter. So even if all of those guys are there, it seems that in the absence of vinculin, none of them seem to be functioning. So not having vinculin is as bad as not having alpha-catenin. And that was what was so surprising for us.
Yeah, in a way it's surprising, yes, because you would expect alpha-catenin anyway to be able to bind actin, but it doesn't.
I mean, stochastically at some point, it doesn't matter. Some level of alpha-catenin should be in the open conformation stochastically, right? Not. It doesn't all, but it's not. It's not.
And the second question is about what you rightly pointed out that both alpha-catenin and vinculin they can change conformation depending on force, right?
Yeah.
So I was wondering if you have ever looked at differences in the alpha-catenin and the vinculin conformation in the different stages you are proposing, so the telogen, anagen, and catagen, and see if depending on the different force that you have in those stages, you would have a different conformation.
It's very cool. Yes, we have looked at that. And in fact, in this stage, so when we go back and, sorry, no, sorry. Let me just, sorry. I was just trying to show you this. So if I go back to this stage, right? So if we look at this growing tip, we see huge amounts of YAP, and we see very low levels of open conformation of alpha-cat.
Yeah.
Okay. Compared to the bulge compartment, which is more quiescent. So the idea is that when the cells are contact inhibited, so we really would love to, we basically believe that this represents a contact inhibited state, and this represents sort of a non-contact inhibited state. And I think the beauty of a system like this is that it's very easy to go from one state to the other simply by modulating the levels of these proteins on the cell surface by simply kicking out some of these transcriptional regulators into the nucleus. So you don't have to do too many things because you can't keep everything totally stuck together. You have to be able to get out of it. So as soon as a signal comes in, you can actually change the junctional dynamics, and that allows for these transcriptional regulators to go into the nucleus and then change the junction itself. And of course, some of the transcriptional some of the genes that are downstream of YAP itself are also junction proteins. And so that sort of, in some sense, would make sense in this kind of regulation. So yes, even though I've only shown you Ki67, this also has very high levels of YAP and low levels of alpha-18.
Yeah, okay, so I would have other questions, but I would like to give space to others to...
Yeah, let's not monopolize all the conversation.
Otherwise, I continue. Please.
I have a very short question here. Very nice talk.
Thank you.
And let's see that, could you modulate the level of YAP? I mean, even inhibiting or overexpressing, and to see any effect in the apparatus that you have there of connectivity between the cells?
Yeah, that is a really interesting question because, is YAP the cause or the consequence of these effects, right?
Exactly.
Yeah, and yes, we also thought long and hard about this. In fact, at some point we were talking to Marius about looking at the YAP knockout, but I would argue that this is a consequence of failed junctions. So in a vinculin knockout, even if you were to reduce the levels of YAP, you would not be able to rescue the junctions because you're not going to be able. So the YAP going into the nucleus is simply a function of alpha-catenin, at least in this instance, not being able to hold on to it. And in some sense, this is very reminiscent of just these cells. So these are the bulk stem cells, and these are the transit amplifying cells. So in the transit amplifying cell, there's lower levels of alpha-cat, there's lower levels of p-cadherin, there's higher levels of YAP. And so our bulk compartment is really functioning more like a transit amplifying compartment. It's no longer functioning as this quiescent, sort of contact inhibited state. And so that is sort of the thesis at the end of all of this. And so it kind of functions like some kind of a stem cell after a fashion. These truly are not stem cells, because if you were to take these cells out of the animal and force them to make hair follicles, they fail to do so. But in this niche, under these context, in this sort of way, it is still able to function somehow to generate a hair follicle, albeit sort of a not normal looking one. So the function of YAP, I mean, the fact that YAP is going to the nucleus, and if we were able to modulate YAP, so we actually did do that. We actually did use inhibitors to get rid of YAP, but that didn't change the junctions in any way.
Okay.